Biotechnology principles and processes. In this chapter, we learn about principles of biotechnology, tools of recombinant DNA technology, processes of recombinant DNA technology. ಬಯೋಟೆಕ್ನಾಲಜಿ ಪ್ರಮುಖವಾಗಿ ಡೀಲ್ ವಿತ್ ಅಂದರೆ ಇದು ಯಾವ ಜೊತೆ ಡೀಲ್ ಮಾಡುತ್ತೆ ಅಂದರೆ ಪ್ರಾಡಕ್ಟ್ಸ್ ಆ್ಯಂಡ್ ಪ್ರಾಸೆಸಸ್ ದಟ್ ಯೂಸ್ಫುಲ್ ಟು ಹ್ಯೂಮನ್ಸ್. And example for this biotechnologies: in vitro fertilization leading to a test tube baby, and synthesizing a gene and using it and developing a DNA vaccine or correcting a defective gene. These are all parts of the biotechnology. And the European Federation of Biotechnology has given a definition of biotechnology. It is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. ಆ್ಯಂಡ್ ಇದರಲ್ಲಿ ಪ್ರಮುಖವಾಗಿ ಮುಖ್ಯವಾದಂಥ ಎರಡು ಮಾಡರ್ನ್ ಬಯೋಟೆಕ್ನಾಲಜಿಯಲ್ಲಿ ಎರಡು ಕೋರ್ಗಳಿದ್ದಾವೆ. ಪ್ರಿನ್ಸಿಪಲ್ ಆಫ್ ಬಯೋಟೆಕ್ನಾಲಜಿ: ದೇರ್ ಆರ್ 2 core techniques that enable birth of modern technology. These are genetic engineering and bioprocess engineering. Genetic engineering ಅಂದರೆ ಆಲ್ಟರ್ ದ ಕೆಮಿಸ್ಟ್ರಿ ಆಫ್ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ as DNA or RNA and thus change the phenotype of the host organism. And next one is bioprocesses engineering. In this, maintenance of sterile microbial contamination free ambience in chemical engineering processes to enable growth of only the desirable microbe or eukaryotic cells. ಅಸೆಕ್ಷುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಏನು ಮಾಡುತ್ತೆ ಅಂದರೆ ಜೆನೆಟಿಕ್ ಇನ್ಫಾರ್ಮೇಷನ್ನ ಕಾಪಾಡುತ್ತೆ. ಆದರೆ ಸೆಕ್ಷುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ ವಿಲ್ ಪರ್ಮಿಟ್ಸ್ ದ ವೇರಿಯೇಷನ್, ಅಂದರೆ ಬೇರೆ ಬೇರೆ ಭಿನ್ನತೆಗಳನ್ನು ಹೊಂದಿಕೊಳ್ಳೋದಕ್ಕೆ ಇದು ಅವಕಾಶವನ್ನು ಮಾಡಿಕೊಡುತ್ತೆ. ಆದರೆ ಅಸೆಕ್ಷುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಇಟ್ ವಿಲ್ ನಾಟ್ ಪರ್ಮಿಟ್ ವೇರಿಯೇಷನ್ಸ್. The genetic engineering include creation of recombinant DNA and use of gene cloning and gene transfer. And piece of DNA would not be able to multiply itself in the progeny cells of the organisms. ಅಂದರೆ ಯಾವುದೇ ಒಂದು ಏಲಿಯನ್ ಪೀಸ್ ಆಫ್ DNA ಅದು ತನ್ನಷ್ಟು ತಾನೇ ಒಂದು ಪ್ರೊಜೆನಿ ಸೆಲ್ಲಲ್ಲಿ ಮಲ್ಟಿಪ್ಲೈ ಆಗಲ್ಲ, ಅನ್ಲೆಸ್ ಇಟ್ ಈಸ್ ಇಂಟಿಗ್ರೇಟ್ ವಿತ್ ದ ಜೀನೋಮ್ ಆಫ್ ದ ರೆಸಿಪಿಯೆಂಟ್. ಅಂದರೆ ಇದನ್ನು ನಾವು ಯಾವಾಗಾದರೂ ಈ ಒಂದು ಏಲಿಯನ್ ಪೀಸ್ ಆಫ್ DNA ನಾವು ಬೇರೆ ಸೆಲ್ನ ಜೀನೋಮ್ ಜೊತೆಗೆ ನಾವು ಇಂಟಿಗ್ರೇಟ್ ಮಾಡಿದಾಗ ಮಾತ್ರ ಇಟ್ ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ಮಲ್ಟಿಪ್ಲೈ ಇಟ್ ಸೆಲ್ಫ್ ಇನ್ ದ ಪ್ರೊಜೆನಿ ಸೆಲ್ಸ್. Because the alien piece of DNA has become part of chromosome, which has the ability to replicate. And in a chromosome, there is a specific DNA sequence called origin of replication, which is responsible for the initiating replication. 1972ರಲ್ಲಿ ಸ್ಟಾನ್ಲೆ ಕೋಹೆನ್ ಮತ್ತು ಹರ್ಬರ್ಟ್ ಬೋಯರ್ ಅವರು ಮಾಲಿಕ್ಯುಲರ್ ಸೀಜರ್ನ ಉಪಯೋಗಿಸಿಕೊಂಡು ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆನ್ಸ್ಗೆ ಕಾರಣವಾಗಿರುವಂಥ ಆ ಜೀನ್ನ ಪ್ಲಾಸ್ಮಿಡಿಂದ ಐಸೋಲೇಟ್ ಮಾಡ್ತಾರೆ. ಆ ಮಾಲಿಕ್ಯುಲರ್ ಸೀಜನ್ ಏನಂತ ಕರೀತಾರೆ ಅಂದರೆ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್ಸ್ ಅಂತ ಕರೀತಾರೆ. ಮತ್ತು ದ ಕಟ್ ಪೀಸ್ ಆಫ್ DNA ವಾಸ್ ದೆನ್ ಲಿಂಕ್ಡ್ ವಿತ್ ದ ಪ್ಲಾಸ್ಮಿಡ್ DNA. ದ ಪ್ಲಾಸ್ಮಿಡ್ DNA ಆ್ಯಕ್ಟ್ ಆಸ್ ಎ ವೆಕ್ಟರ್ಸ್ ಟು ಟ್ರಾನ್ಸ್ಫರ್ ದ ಪೀಸ್ ಆಫ್ DNA ಅಟ್ಯಾಚ್ಡ್ ಟು ಇಟ್. ಆ ಕಟ್ ಮಾಡಿರುವಂಥ ಪೀಸ್ ಆಫ್ DNA ಇವಾಗ ಪ್ಲಾಸ್ಮಿಡ್ DNA ಜೊತೆಗೆ ಲಿಂಕ್ ಮಾಡ್ತಾರೆ. ಆ ಪ್ಲಾಸ್ಮಿಡ್ DNA ಏನಾಗಿ ಆ್ಯಕ್ಟ್ ಮಾಡುತ್ತೆ ಅಂದರೆ ವೆಕ್ಟರ್ಸ್ ಆಗಿ ಆ್ಯಕ್ಟ್ ಮಾಡುತ್ತೆ. Linking of the antibiotic resistant gene with the plasmid vector become possible with the enzyme DNA ligase, which act as on cut DNA molecules and join their ends, and results into a circular autonomously replicating DNA created in vitro and is known as recombinant DNA. Then it is recombinant DNA is transferred into the E. coli bacteria, and it could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistance gene in E. coli was called as a cloning. 3 steps are there in genetically modifying an organism. First one is identification of DNA with desirable genes, introduction of the identified DNA into the host, maintenance of introduced DNA in the host, and transfer of the DNA to its progeny. In the year 1963, the 2 enzymes responsible for restricting the growth of bacteriophage to always cut DNA at specific sequence. It is in 6 base pairs. It is known as recognition sequence. Today we have more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria. And convention for naming these enzymes, the first letter of the name could be genus, second two letters come from species of the prokaryotic cell. Unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. And separation and isolation of DNA fragments. The cutting of DNA by restriction endonucleases result in the formation of fragments DNA. These fragments are separated by using a technique known as gel electrophoresis. DNA fragments are negatively charged molecules. They can be separated by forcing them to move towards anode under an electric effect. Nowadays, the most commonly used matrix is agarose, which is a natural polymer extracted from seaweeds. The DNA fragments separate according to their size through a sieving effect. Sieving effect is nothing but electric effect. The separated DNA fragments we can visualize only after the staining of the DNA with a compound known as ethidium bromide, followed by exposure to UV radiation. And we can see the bright orange color bands of DNA. And elution. Elution means the separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elution. Means the separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This is known as elution. And it is diagrammatic representation means typical agarose gel electrophoresis technique. And cloning vectors. Plasmid and bacteriophage have the ability to replicate within the bacterial cells independent of control of chromosomal DNA. And bacteriophages, because of their high number per cell, have the very high copy number for their of their genome. And some plasmids have only one or two copies, whereas others have 15 to 100 copies per cell. Their numbers can go even higher also. If we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its number equal to the copy number. And the features are required to facilitate cloning into a vector or origin of replication. This is a sequence from where replication starts, and any piece of DNA when linked to the sequence can be made to replicate within the hot cells. ಅಂದರೆ ಯಾವುದಾದ್ರೂ ಒಂದು ಡಿ ಎನ್ ಎ ಪೀಸ್ ಎನಿ ಏಲಿಯನ್ ಪೀಸ್ ಏನಾದರೂ ಲಿಂಕ್ ಆಯಿತು ಅಂದರೆ ಆವಾಗ ರೆಪ್ಲಿಕೇಶನ್ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಆ ಸ್ಪೆಸಿಫಿಕ್ ಸೈಟ್ ಏನಂತ ಕರಿತೀವಿ ಅಂದರೆ ರಿಪ್ಲಿಕೇಶನ್, ಒರಿಜಿನ್ ಆಫ್ ರಿಪ್ಲಿಕೇಶನ್ ಅಂತ ಕರಿತೀವಿ. And the sequence is also responsible for the controlling the copy number of the linked DNA. Selectable marker. In addition to origin of replication, a vector needs selectable markers. Selectable markers are nothing but a gene of plasmid, which helps in identifying and eliminating the non-transformants and selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. And the amplicin, chloramphenicol and tetracycline and vectors for cloning genes in plants and animals. Agrobacterium tumefaciens, a pathogen of several dicot plants, is able to deliver a piece of DNA known as tDNA to transform normal plant cells into a tumor and direct these tumor cells to produce the chemicals required by the pathogens. For example, retroviruses in animals have the ability to transfer normal cells into cancerous cells. And competent host for transformation with recombinant DNA. The bacterial cells must first be made competent to take up DNA. This is done by creating them with a specific concentration of divalent cation such as calcium, magnesium, etc., which increase the efficiency with which DNA enters the bacteria through pores in its cell wall. And next, recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius and then putting them back on ice. These will create pores on the cell walls. And another method is microinjection. The recombinant DNA is directly injected into the nucleus of an animal cell. And another method is suitable for plants, is nothing but cell bombarded with high velocity of microparticles of gold or tungsten coated with DNA in method known as biolistic or gene gun. And last method is known as disarmed pathogen vectors, which when allowed to infect the cell transfer the recombinant DNA into the host. And processes and recombinant DNA technology. Recombinant DNA technology involves several steps. Restriction enzymes belong to a large class of enzymes called nucleases. These are two kinds: exonucleases and endonucleases. Exonucleases removes nucleotides from the ends of the DNA, whereas endonucleases makes cuts at a specific position within the DNA. And these enzymes cut their helix at specific points in their sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. And it is steps in formation of recombinant DNA by action of restriction endonuclease enzyme. ಫಸ್ಟ್ ಏನು ಮಾಡುತ್ತೆ ಅಂದರೆ ಎಂಜೈಮ್ ಕಟ್ಸ್ ಬೋತ್ ದ ಡಿ ಎನ್ ಎ ಸ್ಟ್ಯಾಂಡ್ಸ್ ಸೇಮ್ ಸೈಟಲ್ಲಿ ಎಂಜೈಮ್ ಡಿ ಎನ್ ಎ ಸ್ಟ್ಯಾಂಡ್ನ ಕಟ್ ಮಾಡುತ್ತೆ. ಅಂದರೆ ಎಲ್ಲಿ GAA-TTC ಸೀಕ್ವೆನ್ಸ್ ಇರುತ್ತೋ ಅಲ್ಲಿ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಎಂಜೈಮ್ ಏನಾಗುತ್ತೆ ಕಟ್ ಮಾಡುತ್ತೆ ಡಿ ಎನ್ ಎ ಸ್ಟ್ಯಾಂಡ್. ಆಮೇಲೆ ಫಾರಿನ್ ಡಿ ಎನ್ ಎ ಸ್ಟಿಕೇನ್ಸ್ ಮತ್ತು ವೆಕ್ಟರ್ ಡಿ ಎನ್ ಎ ಸ್ಟಿಕೇನ್ಸ್ನ ಜಾಯಿನ್ ಮಾಡಲಾಗುತ್ತೆ. ಯಾವುದ್ರ ಮೂಲಕ ಅಂದರೆ ಇವೆರಡನ್ನೂ ಕೂಡ ಡಿ ಎನ್ ಎ ಲೈಗೇಸ್ ಹೆಲ್ಪ್ ತಗೊಂಡು ವೆಕ್ಟರ್ ಡಿ ಎನ್ ಎ ಸ್ಟಿಕೇನ್ಸ್ ಮತ್ತು ಫಾರಿನ್ ಡಿ ಎನ್ ಎ ಸ್ಟಿಕೇನ್ಸ್ನ ಜಾಯಿನ್ ಮಾಡ್ತೀವಿ. ಆ ಡಿ ಎನ್ ಎ ನಾವು ಏನಂತ ಕರಿತೀವಿ ಅಂದರೆ ರಿಕಾಂಬಿನೆಂಟ್ ಡಿ ಎನ್ ಎ ಅಂತ ಕರಿತೀವಿ. ಸ್ಟಿಕೇನ್ಸ್ ಮೀನ್ಸ್ ವೆನ್ ದ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಎಂಜೈಮ್ ಕಟ್ಸ್ ದ ಡಿ ಎನ್ ಎ ಸ್ಪೆಸಿಫಿಕ್ ಸೈಟ್, ದ ರಿಮೈನಿಂಗ್ ಪಾರ್ಟ್ ಆಫ್ ದ ಓವರ್ ಹ್ಯಾಂಗಿಂಗ್ ಸ್ಟ್ರೆಚಸ್ಸನ್ನು ಏನಂತ ಕರಿತೀವಿ ಅಂದರೆ ಸ್ಟಿಕೇನ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಪಾಲಿಂಡ್ರಮಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ಅಂತಂದರೆ ನಾವು ಹಿಂದೆಯಿಂದ ಅಂದರೆ ಲೆಫ್ಟ್ ಸೈಡಿಂದ ಓದಿದ್ರು ರೈಟ್ ಸೈಡಿಂದ ಓದಿದ್ರು ಒಂದೇ ಅರ್ಥ ಕೊಡಬೇಕು. ಇವಾಗ ಮಲಯಾಳಂ ಇದನ್ನು ಲೆಫ್ಟ್ ಸೈಡಿಂದ ಓದಿದ್ರು ಮಲಯಾಳಂ ಅಂತಲೇ ಮೀನಿಂಗ್ ಕೊಡುತ್ತೆ, ಹಾಗೆ ರೈಟ್ ಸೈಡಿಂದ ಓದಿದ್ರು ಮಲಯಾಳ ಅಂತಲೇ ಮೀನಿಂಗ್ ಕೊಡುತ್ತೆ. ಯಾವಾಗ ಎಂಜೈಮ್ಸ್ನ ಹೆಲ್ಪ್ ತೊಗೊಂಡು ನಾವು ಡಿ ಎನ್ ಎನ ಕಟ್ ಮಾಡ್ತೀವೋ ಆವಾಗ ಉಳಿದಿರುವಂಥ ಪಾರ್ಟ ನಾವೇನಂತ ಕರಿತೀವಿ, ಸ್ಟಿಕೇನ್ಸ್ ಅಂತ ಕರಿತೀವಲ್ವಾ, ಹೀಗೆ ಅದೇ ಕೊಟ್ಟಿದ್ದಾರೆ. ದೇರ್ ಆರ್ ಓವರ್ ಹ್ಯಾಂಗಿಂಗ್ ಸ್ಟ್ರೆಚಸ್ ಕಾಲ್ಡ್ ಸ್ಟಿಕೇನ್ಸ್ ಆನ್ ಈಚ್ ಅದರ್ are named so because they form hydrogen bonds with their complementary cut counterparts. ಏನಕ್ಕೆ ಅಂತಂದರೆ ಆ ರಿಮೂವ್ ಆದಂಥ ಪಾರ್ಟ್ಗಳು ತಮ್ಮ ಕಾಂಪ್ಲಿಮೆಂಟ್ರಿ ಸ್ಟ್ಯಾಂಡ್ಸ್ ಅಂದರೆ ಸೀಕ್ವೆನ್ಸ್ ಜೊತೆಗೆ ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್ನ ಫಾರ್ಮ್ ಮಾಡಿಕೊಂಡಿರ್ತಾವಲ್ಲ, ಅದು ರಿಮೂವ್ ಮಾಡಿದ್ಮೇಲೆ ಅವ್ರ ಎಂಡ್ಸ್ ಏನಾಗುತ್ತೆ ಸ್ಟಿಕ್ಕಿ ಆಗಿರುತ್ತೆ. ಅದಕ್ಕೆ ಅದನ್ನು ಸ್ಟಿಕ್ಕಿ ಎಂಡ್ಸ್ ಅಂತ ಕರೀತಾರೆ. ಈ ಸ್ಟಿಕ್ಕಿ ಎಂಡ್ಸ್ the stickiness of the ends facilitates the action of the enzyme DNA ligase. ಸ್ಟಿಕ್ಕಿ ಎಂಡ್ಸ್ ಇರ್ತಾವಲ್ಲ ಅವು ಡಿ ಎನ್ ಎ ಲೈಗೇಸ್ ಫಂಕ್ಷನ್ನ ಇನ್ನೂ ಸುಲಭವಾಗಿ ಮಾಡಿಕೊಡುತ್ತೆ. ಮೇನ್ಲಿ RNAs ಆರ್ ಯೂಸ್ಡ್ ಇನ್ ಜೆನೆಟಿಕ್ ಇಂಜಿನಿಯರಿಂಗ್ ಟು ಫಾರ್ಮ್ ರಿಕಾಂಬಿನೆಂಟ್ ಮಾಲಿಕ್ಯೂಲ್ ಆಫ್ DNA. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and these can be joined together end to end using DNA ligases. And it is a diagrammatic representation of recombinant DNA technology. First, foreign DNA and vector DNA. These both are cut by using same restriction endonuclease enzymes at specific point. And after ligases join foreign DNA to plasmid. This is called as recombinant DNA molecule. Next, transformation. Cells divide and it will be replicate. ಫಸ್ಟ್ ಫಾರಿನ್ ಡಿ ಎನ್ ಎನ ತೊಗೊತಾರೆ ಮತ್ತು ವೆಕ್ಟರ್ ಡಿ ಎನ್ ಎನ ತೊಗೊಂಡು ಈ ಎರಡು ಡಿ ಎನ್ ಎನ ಸೇಮ್ ಎಂಜೈಮ್ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಸ್ಪೆಸಿಫಿಕ್ ಸೈಟಲ್ಲಿ ಕಟ್ ಮಾಡ್ತಾರೆ. ಆ ಕಟ್ಟಾಗಿರುವಂಥ ಒಂದು ಫಾರಿನ್ ಡಿ ಎನ್ ಎನ ವೆಕ್ಟರ್ ಡಿ ಎನ್ ಎಯಲ್ಲಿ ಬೈ ಯೂಸಿಂಗ್ ಲೈಗೇಸನ್ನು ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಜಾಯಿನ್ ಮಾಡ್ತಾರೆ. ಅದನ್ನು ನಾವೇನಂತ ಕರಿತೀವಿ ಅಂದರೆ ರೀಕಾಂಬಿನೆಂಟ್ ಡಿ ಎನ್ ಎ ಮಾಲಿಕ್ಯೂಲ್ ಅಂತ ಕರಿತೀವಿ. ಅದನ್ನು ಆ ವೆಕ್ಟರನ್ನು ನಾವು ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡ್ತೀವಿ, ಸೆಲ್ಸ್ ಡಿವೈಡ್ ಆಗುತ್ತೆ, ಹಾಗೆ ಆ ಒಂದು ಡಿ ಎನ್ ಎ ಕೂಡ ಡಿವೈಡ್ ಆಗಿ ಡಿಸೈರಬಲ್ ಕ್ಯಾರೆಕ್ಟರ್ನ ಕೊಡುತ್ತೆ. And unless one cuts the DNA vector and the source of DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. ಅಂದರೆ ವೆಕ್ಟರ್ DNA ಮತ್ತು ಇನ್ಸರ್ಷನ್ ಆಫ್ recombinant DNA into the host cell or organism. There are several methods of introducing the ligated DNA into recipient cells. Recipient cells, after making them competent to receive, if a recombinant DNA bearing a gene of resistant to an antibiotic, example amplicin, is transferred into E. coli cells, the host cell became transferred into amplicin resistant cells. The amplicin resistance gene is, in this case, is called as a selectable marker. Obtaining the foreign gene product. The ultimate aim is to produce a desirable protein. After having cloned the gene of interest and having optimized the conditions to induce the expression of the target protein, one has to consider producing it on a large scale. If any protein encoding gene is expressed in a heterologous host, it is called as a recombinant protein. The cells of harboring a cloned gene of interest may be grown on a small scale in the laboratory. The culture may be used for extracting the desired protein and then purifying it by using different separation techniques. Small volume cultures cannot yield appreciable quantities of products. To produce large quantities, the development of bioreactors where the large volumes 100 to 1000 liters of culture can be processed was required. Bioreactors can be thought of as a vessels in which raw materials are biologically converted into specific products, individual enzymes, etc., using microbial, plant, animal, or human cells. A bioreactor provides the optimal conditions for achieving the desired product by providing optimum growth conditions, temperature, pH, substrate, salts, vitamins, oxygen. And it is diagram of simple sterile tank bioreactor and spag sterile tank bioreactor. And a sterile tank reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor contents. The stirrer facilitates the even mixing and oxygen availability through the bioreactor. Alternatively, air can be bubbled through the reactor. And bioreactor is an agitator system, an oxygen delivery system, and foam control system, a temperature control system, pH control system, sampling ports so that small volume of the culture can be withdrawn periodically. Downstream processing. After completion of biosynthetic stage, the product has to be subjected to the series of processes before it is ready for marketing as finished product. The processes include separation and purification, which are collectively referred to as downstream processing. The product has to be formulated with suitable preservatives. Such formulation has to undergo through clinical trials, as in case of drugs. Strict quality control testing for each product also be required. The downstream processing and quality control testing vary from product to product.